Welcome to today's lecture. So today we'll be talking about metabolism and absorption of vitamin B12, also called cobalamin. Now, vitamin B12 is one of the water-soluble vitamins, but it has a unique characteristic, and that is the fact that its absorption is specific to the ileum. So the intestine, the small intestine, is divided into three segments: the duodenum, the jejunum, and the ileum. So cobalamin absorption occurs only at the ileum. Now, cobalamin consists of 4 pyrrole rings that surrounds a cobalt atom. Okay, consists of 4 pyrrole rings that surrounds a cobalt atom. So, of course, the symbol is represented as cobalt, CO. Now, this is the natural form of vitamin B12. So, the natural form of vitamin B12 is cobalamin, which consists of 4 pyrrole rings, okay, that surrounds a cobalt atom. But there are several synthetic forms. When a cyanide group gets attached to the cobalt atom, it will form cyanocobalamin, as a cyanide group, okay? And then an hydroxyl group gets attached to the cobalt atom, it will form hydroxycobalamin. So this is hydroxyl group. Then when a sulfate group gets attached to the cobalt atom rather than either of these, so for a sulfate group, it will be sulfatocobalamin. And then when a chloride group gets attached to the cobalt atom, of course, it's going to be chlorocobalamin. Also, when a nitride group gets attached, okay, it's going to form nitritocobalamin. Now, all these forms are synthetic forms, and the reason for this synthetic form is for crystallization of cobalamin. Among all these several forms, the most potent form is the hydroxycobalamin, especially when it is taken orally. The most potent form is the hydroxycobalamin, and that is because it lasts longer in the body system. When taken orally, it lasts longer, and also it has the ability to withstand enzymatic reactions compared to the other forms. And based on these two reasons, it is more potent. There are several sources of vitamin B12, but mainly it is obtained from animal proteins or from animal products, and that include fish, liver, egg, red meat, poultry, dairy products, milk, cheese, and yogurt. Now, for the absorption of vitamin B12, several components are required. The first is transcobalamin. Now, transcobalamin is a protein that is secreted from the acinar cells of the salivary gland. That's for transcobalamin. It's a protein secreted from the acinar cells of the salivary gland. The next one is hydrochloric acid, and then we have the intrinsic factor. Now, the hydrochloric acid and the intrinsic factor are secreted by parietal cells. Parietal cells of the stomach. So in the stomach, there are parietal cells, also called ozontic cells. So these cells are responsible for the secretion of hydrochloric acid and intrinsic factor. And these parietal cells are usually found at the fundus or the body of the stomach. So we have something like this. This is the cardia. This is the fundus. And then this is the body. So the parietal cells or the ozontic cells are usually found at the fundus or the body of the stomach. Sometimes it cannot be found, it can also be found at the cardia, and they are responsible for the secretion of hydrochloric acid and intrinsic factor. Another name for this transcobalamin one is cobalamin. Cobalamin, which can also be called arrow binder. And the next one is pepsin. The sheath cells secrete pepsinogen. But then this pepsinogen is an inactive form that needs to be converted to pepsin, which is the active form. Now, for this pepsinogen to be converted to pepsin, it must be acted upon by hydrochloric acid. So the hydrochloric acid that has been secreted by the parietal cells will act on pepsinogen to form pepsin. Hydrochloric acid. Another one is trypsin. Now, trypsin is also obtained from the conversion of trypsinogen, and trypsinogen is secreted by the acinar cells of the pancreas. So this is a diagram of the stomach and then the duodenum. This is the duodenum. Somewhere around there is the pancreas. Okay, and then there are what we call acinar cells. These acinar cells secrete trypsinogen. Okay, trypsinogen into the duodenum. Now, in the duodenum, there are enzymes called enteropeptidases or enterokinases. Now, these enzymes act on the trypsinogen that has been secreted by the acinar cells of the pancreas, right? In the duodenum, these enteropeptidases act on trypsinogen and converts it to trypsin. Acted upon by what? Enteropeptidases. The next one is calcium ion and releasing factors. Now, calcium ion and releasing factors are also secreted at the duodenum. Okay, and then it is carried, it flows along until it gets to the ileum where cobalamin is absorbed. The next one is cobalamin specific receptor. This can also be called the cobalamin. So, okay, cobalamin specific receptor can also be referred to as the Cobalamin. So this is a receptor that is present at the ileum where cobalamin is absorbed. This receptor is specific to cobalamin, where cobalamin binds to this receptor and then its absorption is enhanced. The next one is transcobalamin 2. Now, transcobalamin 2 is usually in the circulation. So after the absorption of cobalamin, transcobalamin 2 gets bound, okay, or binds to cobalamin and then transports it to the liver. For the absorption of cobalamin, I'll be talking about when it is obtained from the food and also when it is obtained in synthetic form. So both the synthetic form and the one obtained from food, they have similar mode of metabolism and absorption, but just a little difference. So first of all, let us talk about the absorption of cobalamin that is obtained from the diet. Now, I have some symbols that represents some of these components that are required. This represents the animal protein. This here represents cobalamin. This represents transcobalamin 1. This represents intrinsic factor. This represents cobalamin, that is the cobalamin specific receptor. And then this represents transcobalamin 2. This is cobalamin attached or bound to animal protein. So when cobalamin is obtained from the diet, because it's obtained from animal protein, the animal protein is bound together with the cobalamin. And then when it gets into the mouth, the acinar cells secretes transcobalamin 1. So this is transcobalamin 1. Now, transcobalamin 1 is supposed to bind to cobalamin and transport it. But because cobalamin is already bound to the animal protein, transcobalamin 1 will not be able to bind to it in the mouth. So it's going to follow the complex. This is the animal protein cobalamin complex. So this is animal protein cobalamin complex. So when this complex moves down through the esophagus, the transcobalamin 1 will follow it, okay? Will be going down with it. And then on getting to the stomach, this is the Schiff cell. The Schiff cell will secrete pepsinogen, okay? So P represents pepsinogen. And then the parietal cell will secrete hydrochloric acid and intrinsic factor. Now, the hydrochloric acid that is secreted by the parietal cell, following this arrow, will act on the animal protein to digest it. So when it digests it, HCl will also act on pepsinogen secreted by the Schiff cells and convert it to pepsin. The pepsin, which is the activated form of the pepsinogen, will now further act on the animal protein to degrade it or to hydrolyze it. When that occurs, the cobalamin will become free. Now, as you can see here, transcobalamin 1 is still accompanying the complex. So immediately cobalamin becomes free, transcobalamin 1 will bind to cobalamin. Now, the intrinsic factor secreted by the parietal cell also is supposed to bind to cobalamin and ensures that it is being absorbed, because it is the intrinsic factor that will carry it to where it's going to be absorbed. But because the transcobalamin 1 is already bound to cobalamin, the intrinsic factor will not be able to bind to cobalamin. Rather, it's also going to follow the complex. So now, this is the transcobalamin 1. So this is a transcobalamin 1 cobalamin complex. So the intrinsic factor will also follow this complex down into the duodenum. Now, on entering into the duodenum, this is the pancreas, okay? The acinar cells of the pancreas will also secrete trypsinogen, okay? It will secrete trypsinogen into the duodenum. Now, trypsinogen is the inactivated form. So, inside the duodenum, there are enzymes called enteropeptidases that will act on the trypsinogen and convert it to trypsin, which will become the active form. Now, the active form, which is trypsin, will now hydrolyze this transcobalamin 1. Okay? And once this transcobalamin 1 is being hydrolyzed, you can see that cobalamin again will be free. But because this intrinsic factor has been following it and needs to be bound to cobalamin, immediately it is free. The intrinsic factor will immediately bind to the cobalamin. So this is the intrinsic factor cobalamin complex. Okay, now this complex will now move. This is the duodenum into the jejunum and then to the ileum. Okay, so once it gets to the ileum, there is a receptor that is specific to cobalamin, which is called the Cobalamin. So this is the receptor. You can see the receptor here. And then also calcium ion and releasing factors that are produced at the duodenum will flow along with this complex to the point where the receptor that is specific to cobalamin is located. So when it gets to that point, the releasing factor and the calcium ion will now enhance the release of cobalamin from the intrinsic factor. Okay, so this releasing factor and the calcium ion will enhance the release of cobalamin from the intrinsic factor, and then cobalamin will become bound to the its specific receptor. After being bound into the specific receptor, it will be absorbed into the circulation. Now this is the circulation, this is the blood. Okay, so let's say this is the circulation, the blood. Now after its absorption, it will now get bound to transcobalamin 2, okay, which is also a protein in the circulation. Now that transcobalamin 2 will now carry the cobalamin to the liver. So this is the transcobalamin 2 cobalamin complex. Now, this explains the absorption of cobalamin that is obtained from food, from animal protein. What about the synthetic form? How is it absorbed? It is similar. The difference is the absence of this animal protein. So, in a synthetic form, when cobalamin is taken in the synthetic form, it is taken in as a free cobalamin, not bound to the animal protein. So, getting into the mouth, it is also free, okay? Then, the acinar cells will secrete transcobalamin 1. So, from here, transcobalamin 1 will get bound, okay? Transcobalamin 1 will get bound to cobalamin. Instead of the presence of this animal protein, transcobalamin 1 will get bound because this animal protein is not there. Because cobalamin is obtained in the synthetic form, it is free, it is not bound to any animal protein. So there is no animal protein here, there is no animal protein here. So cobalamin here will be free. And because it is free, the transcobalamin 1 that will be secreted by the acinar cells will get bound. Okay, so when it gets bound, it will now move along into the stomach, and then in the stomach, it will also not change. Okay, so it will move directly from the mouth after the binding of transcobalamin 1 to cobalamin. It will move down through the esophagus into the stomach and then to the duodenum, small intestine, to the duodenum. It is in the duodenum that trypsin will now act on the transcobalamin 1 and then dissociate. So for a synthetic form of cobalamin that is taken in from the mouth, transcobalamin 1 binds to cobalamin to form transcobalamin 1 cobalamin complex. Then that complex will move from the esophagus to the stomach and then directly to the duodenum. So all this process of digestion and all of that will not occur because there is no animal protein that is bound to cobalamin. So what are the functions of this vitamin B12? In my previous class, I stated that cobalamin acts as a coenzyme to dissociate 5-methyl tetrahydrofolate to produce tetrahydrofolate, which is the active form or the biologically active form of folate. So we have something like this. (5-methyl tetrahydrofolate, cobalamin, to give rise to methylcobalamin, and tetrahydrofolate. So in my previous class, I stated that cobalamin acts as a coenzyme to dissociate 5-methyl tetrahydrofolate to producing methylcobalamin and tetrahydrofolate, and that this tetrahydrofolate is the biologically active form of folate, and that for folate to act or to carry out its physiological role, it actually does that in this form. And then I also stated that this tetrahydrofolate now goes to function in the 1-carbon metabolism, where there is a transfer of 1 carbon unit between molecules, and that 1 carbon metabolism results in DNA synthesis in the bone marrow. Okay, so this ensures DNA synthesis in the bone marrow, and because there is DNA synthesis in bone marrow, there will be normal proliferation and maturation of red blood cells. That is to say, if there is deficiency of cobalamin, okay, if there is deficiency of cobalamin, it means 5-methyl tetrahydrofolate will not dissociate to form methylcobalamin and tetrahydrofolate, and so the function of tetrahydrofolate will be inhibited. That is to say, 1 carbon metabolism cannot take place, and when 1 carbon metabolism cannot take place, it means DNA synthesis in the bone marrow will be inhibited, and when DNA synthesis in the bone marrow is inhibited, it will result in anemia. So the functions of cobalamin include DNA synthesis, which of course will help in normal proliferation and maturation of red blood cells. So this will include erythropoiesis. It also plays a role in maintaining the nervous system, maintenance of the nervous system. And of course, several other functions. Now, that is to say, if there is deficiency of cobalamin, it means DNA synthesis in the bone marrow cannot take place. And that is to say, erythropoiesis cannot take place. And that will result, so this is deficiency, okay, of vitamin B12. And that will result in anemia. And the type of anemia is megaloblastic anemia. That is because normally DNA synthesis in the bone marrow will enhance the normal proliferation and maturation of red blood cells, and then they are released into the circulation. But when DNA synthesis is inhibited as a result of deficiency of cobalamin, it means the normal proliferation and maturation of red blood cells will not occur, and that will result in production of megaloblasts, which are larger in size and have larger nuclei and are also immature. Okay, so normally these megaloblasts are supposed to undergo proliferation to form smaller mature red blood cells, which will be released into the circulation. But because of the inhibition of DNA synthesis as a result of cobalamin deficiency, megaloblasts will no longer undergo that normal proliferation, and so these megaloblasts will be released from the bone marrow into the circulation. Although when they get into the circulation, they may undergo maturation and normal proliferation to produce normal red blood cells, but not enough of these can be formed, not enough mature red blood cells can be formed, and as a result of that, it will result in anemia. The second one is because it helps in the maintenance of the nervous system. If there is deficiency of cobalamin, it will lead to neuropathy, okay, disease conditions that affect nervous system. It will lead to neuropathy, of course, which will include the numbness and tingling of the hands and feet. In my previous class, I also stated that methylcobalamin we have to combine with homocysteine, and that this will result in the production of what? There will be a transfer of this methyl group to homocysteine to form methionine, while cobalamin will go back into the circulation to continue the process, and that this methionine is a precursor, okay, to S-adenosyl methionine, which also stated that this S-adenosyl methionine functions in the transfer of methyl group to several molecules. Now, if there is deficiency in cobalamin, apart from the fact that tetrahydrofolate, which enhances 1-carbon metabolism, will not be formed, methionine also will not be formed, but then there is homocysteine. So, because methionine cannot be formed, there will be accumulation of this homocysteine, okay? There will be an increased concentration of this homocysteine. And from studies, the accumulation of homocysteine has been known to affect or to damage the arteries, and that is an increased risk factor for cardiovascular diseases. So, another condition that can be induced as a result of deficiency of cobalamin is increased risk for developing cardiovascular diseases. Several factors can cause vitamin B12 deficiency. And what are these factors? We have lack of vitamin B12 in diet. Because vitamin B12 is obtained from animal products, strict vegetarians are prone to developing cobalamin deficiency. So another factor that can cause vitamin B12 deficiency is diseases of the salivary gland. So where there is a disease of the salivary gland that can cause a blockage of the salivary gland such that transcobalamin 1 cannot be secreted into the mouth. Such a condition can bring about a deficiency in vitamin B12. For example, benign salivary tumors, okay? So you can see benign for example, benign salivary tumors. Okay, so this is an example of a disease of the salivary gland that can bring about vitamin B12 deficiency. Another factor is autoimmune disease of the digestive tract. So if there is an autoimmune disease, for example, an autoimmune disease that attacks the parietal cells such that intrinsic factor, intrinsic factors are not secreted in sufficient quantity. Also, hydrochloric acid is not secreted in sufficient amount. That condition, okay, can lead to vitamin B12 deficiency. And that thing is disease of the pancreas. Okay, the pancreas has a major role to play. Okay, it secretes the acinar cells of the pancreas secretes pepsinogen into the duodenum. And so if there is a disease of the pancreas such as pancreatitis or cancer of the pancreas, of course the pancreas will not function effectively, and that can as well lead to vitamin B12 deficiency.